This presentation covers Part 1 of Chapter 14, Integration of Nervous System Functions. In this presentation, we will differentiate between sensation and perception, compare the general senses and special senses, learn the different sensory receptors, and define receptor potential and adaptation. Let's begin by covering sensation and perception. Sensation occurs when stimuli activate sensory receptors, which generate action potentials along sensory neurons to the spinal cord and brain. It is the afferent information being sent towards the central nervous system. Sensation occurs in the peripheral nervous system. Perception is the conscious awareness of sensations. Perception occurs when the brain interprets the action potentials generated from sensation. Perception occurs in the cerebral cortex. The image shows sensation as visual receptors being activated, and perception is the brain knowing what it was looking at. Now we will learn about general and special senses. When you were young, you likely learned that we have 5 senses and that they were seeing, hearing, tasting, smelling, and touch. Well, it's time to relearn what you thought was true. Our senses are divided into 2 main groups, general senses and special senses. Let's focus on the general senses first. General senses have receptors all over the body. General sense receptors generate action potentials called generator potentials, which travel towards the brain. General senses are divided into 2 groups, somatic senses and visceral senses. Somatic senses provide information about the skin, muscles, and joints. Somatic senses include touch, temperature, pressure, pain, and proprioception. Proprioception is knowing your body's position and movement. I remembered the somatic senses by TTPPP, touch, temperature, pressure, pain, proprioception. Visceral senses give us information about our internal organs, which are also known as the viscera. Your visceral senses give you information about pain and pressure of your organs. So remember, general senses have receptors all over the body and give us information about touch, pressure, pain, temperature, and proprioception. Now let's learn about the special senses. Special senses have receptors that are located in specific organs, so they are not found all over the body. The 5 special senses are smell, taste, vision, hearing, and balance. Make special note that balance is a special sense. Touch is not a special sense. Special senses are covered in detail in Chapter 15. Now we need to learn the sensory receptors. We can classify sensory receptors in 3 ways: by the type of stimulus they detect, their location in the body, and their structure. Here we have sensory receptors classified by the types of stimuli they detect. Mechanoreceptors detect compression, bending, and stretching of cells. Touch, pressure, proprioception, hearing, and balance all use mechanoreceptors. Chemoreceptors detect chemicals. They are used in taste and smell. Thermoreceptors detect changes in temperature. Photoreceptors detect light energy and are used in vision. Nociceptors detect pain, which is extreme mechanical, chemical, or thermal stimuli. When classifying sensory receptors by location, we have three groups. Cutaneous receptors are found in the skin. Visceral receptors are found in organs. And proprioceptors are found in joints and tendons where they detect stretch and the force of muscle contraction. There are 8 types of general sensory receptors based on their structure. These 8 do not include the special sensory receptors, which we're going to discuss in chapter 15. So let's go over each of these 8. This slide shows 6 of them. The receptor with the simplest structure, and it is also the most common receptor, is free nerve endings. These receptors are similar to dendrites. They are branched and they do not have a capsule. Free nerve endings respond to pain, temperature, itch, joint movements, and proprioception. They include both cold and warm receptors, which detect different temperatures. At a comfortable temperature, both the cold and warm receptors are activated. The second type of receptor is the Merkel disc. It has a flat expansion at the axon terminal. Each flat part is a Merkel disc. They detect light touch and superficial pressure. Notice in the image that they are located close to the surface of the skin. The third receptor is the hair follicle receptor, and they are located around hair follicles in the dermis. These receptors are made of several neurons whose branching dendrites have receptive fields that overlap. This allows for the sensitivity to detect light touch and a slight bending of the hair. Pacinian corpuscles are located deep in the dermis or in the hypodermis, and they detect deep pressure, vibration, or they are associated with joints to detect proprioception. These receptors are onion-shaped and have a capsule of several cell layers that surround a single dendrite or neuron. Meissner corpuscles are made of several branches of a single axon that are associated with specialized Schwann cells. They are surrounded by a connective tissue capsule. Meissner corpuscles are located in the dermal papilla, and they are involved in two-point discrimination. Two-point discrimination is the ability to detect simultaneous stimulations as two separate points. Meissner corpuscles also detect the texture of objects. We have numerous Meissner corpuscles on the tongue and on our fingertips, but fewer on the back. Ruffini end organs are made of branching axons with numerous small terminal knobs surrounded by connective tissue that forms a capsule. They respond to continuous touch or pressure and to depression or stretch of the skin. They are found in the dermis primarily in the fingers. Muscle spindles are made of 3 to 10 specialized skeletal muscle cells. They provide information about the length of the muscles and are involved in maintaining tone as well as being involved in the stretch reflex. Golgi tendon organs are involved in proprioception and are located on a tendon near where the tendon and muscle meet each other. Golgi tendon organs are branched and have small swellings at the ends. The whole receptor is enclosed by a delicate connective tissue covering. This is a table from your textbook that you may find helpful for your studying, so I've included it here. Our final topics are receptor potential and adaptation. Sensory receptors respond to stimuli with a graded potential that is called a receptor potential. If the receptor potential reaches threshold, then an action potential is generated down the axon. Primary receptors work this way, meaning an action potential is generated in response to the receptor potential. Most sensory receptors are primary receptors. Secondary receptors work a little differently. In this case, the receptor cell releases a neurotransmitter that binds to a receptor on an adjacent neuron. This causes a receptor potential in that adjacent neuron. Receptors for smell, hearing, taste, and balance are secondary receptors. Adaptation is the term that describes decreased sensitivity to a continued stimulus. An example is when you first sit down in the library and you easily smell somebody's perfume. After being there a while, you no longer smell the perfume. You have become adapted to that stimulus. We categorize receptors on how easily they adapt to stimuli. Tonic receptors adapt very slowly. Merkel discs and Ruffini ending organs are considered tonic receptors. Phasic receptors adapt rapidly, and they are more sensitive to changes in the stimuli. Pacinian and Meissner corpuscles are phasic receptors. In this presentation, we differentiated between sensation and perception, compared general and special senses, learned the sensory receptors, and defined receptor potential and adaptation.